So as John mentioned yesterday, I work in genetics and genomics, and this is a field that's absolutely exploding with new technologies and methods. In just the last 3 or 4 years, there's probably half a dozen new sequencing technologies that have been introduced that do multiple orders of magnitude higher throughput sequencing and allow you to do different types of samples and different types of sequences, and to do literally millions of sequences in a single run of an instrument. So it's changing the way we think about doing genetics, and that's what I've been talking about a little bit today. It was initially termed next generation sequencing when these new technologies were first introduced, and now they've actually gone on to saying, well, that was just second generation. We're on to the next, the third and fourth generations, but I still call it next generation sequencing. Okay, so sorry about this one. Okay, I work at the MorinPanel Genetics Group at South Florida Science Center, and just to give you a little background, our group focuses on using genetics to understand population structure and taxonomy of cetaceans. So we look at this at several different levels. That population structure is our primary interest within the North Pacific. We use a number of genetic tools for each of these different types of studies that we do, including mitochondrial DNA control region sequencing. That's been something we've used over the last couple of decades, really using between 500 and 1,000 base pairs of control region. That's because that was about what we could easily sequence out of the mitochondrial genome, and in many cases gave us enough information to be able to understand population structure and even taxonomy of different species. We also use microsatellites. They're useful for population structure and individual identification or genetic marker capture. And more recently, we've been developing single nucleotide polymorphisms. These are individual mutations in the DNA sequence, and they're ubiquitous through the genome. And technologies come along to allow us to discover and genotype more of those. I'm not going to go into detail about the different types of markers. I just want to put these up here because you're going to be seeing these terms for the rest of the presentation. We've recently done a lot more at the level of taxonomy and phylogenetics, and that's because even though we're studying, for example, killer whales in the North Pacific, we need to understand not only what our populations are, but how they relate to the species worldwide. And in many cases with cetaceans, we have no idea what the worldwide taxonomy of a cetacean species is. So we're using genetic methods to try to get a better grip on what's out there and then how it's distributed. So I'm going to overview the study on killer whales that we did last year. It was published in July last year, where we looked at mitochondrial genomes of killer whales globally. This was a very collaborative study with people at a bunch of different labs. So most of you must know that there are a variety of different types of killer whales, and they've been called ecotypes typically in the North Pacific, where they don't differ morphologically a whole lot, but there's obvious differences in their dietary specialization, behaviors, and distributions, though they all overlap to some extent in their geographic distributions. So without the ability to have lots of skeletons to measure and ways to look at the detailed morphology, it's hard to say how do you call different species when you're working with photographs and just observations in the field. So with the North Pacific, the ecotypes have been mostly called populations or ecotypes. In the Antarctic, more recently, there have been a number of different types discovered, also described based on their dietary specialization, but they have more morphological differences. And this slide is what John put up yesterday, but type A, B, and C in the Antarctic, and now there's a type D and possibly an E and a B sub A. There are a lot of different types that Bob Pittman's down trying to document right now and get me samples from. But these ones have more morphological differences, and they've suggested, based on a little bit of genetic data and the morphology and behavioral differences, that there might be different species. And then in the North Atlantic recently, my colleague Andy Foote has also described 2 different types. And so there's probably a lot of different ecotypes worldwide. But when we started to do the genetics over 10 years ago, a number of people started looking at using the usual tools, control region, microsatellites, and there was just basically no resolving power. This is a control region phylogeny of killer whales worldwide, done by Hosel et al. 9 years ago. And you can see that this tree is basically one long branch with little sticks sticking out of it. So there's no phylogenetic information here. This is a 1,000 base pair, so it's the entire control region. We only found 13 haplotypes throughout the world in over 100 samples. And there's really no phylogeographic pattern and very small differences between the ecotypes. So the evolutionary relationships of the different ecotypes is not resolved with these data. That's the control region there, the 1,000 base pairs, but the mitochondrial DNA is actually this 16,400 base circular genome, and it has a number of different types of genes in it. And it's a tool that we've used a lot for a lot of reasons. I won't go over that. It's a good tool for understanding phylogenetics and phylogeography and even population structure. But when there's that little information in this little region, the logical question is what would we get if we got more information? So with the ability to do next generation sequencing, we were offered the chance to sequence whole mitochondrial genomes for up to about 150 individuals through a collaboration with one of the companies that markets the technology. And so we used a long-range PCR method, which requires that you have fairly good quality DNA, and PCR amplified fragments of between 5 and 10 kilobases each to cover the entire mitochondrial genome. And then we pool individuals and send them off to the company to do the sequencing. And I'm going to get right to the results in the interest of time, but I'll get back to it and talk about this technology a little bit more in a second. The previous tree, as you remember, was a straight line with a bunch of little sticks on it for individual haplotypes. This is the tree from the mitochondrial genomes. And starting with the North Pacific, up at the top here are the residents and offshores. And they form 2 reciprocally monophyletic groups that show that they are unique, evolutionarily distinct groups, and they're most closely related to each other. But the first surprise, as John said yesterday, is that the transients are way down here in this really deep branch that actually includes all other killer whales worldwide in this cluster, and the transients down here. So that was our first surprise, and in fact, using the technology with this much sequence data, we were actually able to estimate the date of divergence with a pretty good accuracy, and that date is about 700,000 years ago, which is far earlier than had previously been estimated based on other data. The date for the residents and offshores was estimated about 175,000 years ago. And then in the Antarctic, down here we have the A's, B's, and C's. And again, the B's and C's, those are the 2 that are associated with ice and have the different morphology, the different size eye patches. The C type is a dwarf form, and they're most closely related to each other. But again, they're reciprocally monophyletic, and then here's the A types here. And that divergence is estimated about 335,000 years ago and 150 respectively. And then in the North Atlantic, the picture is a little less clear, where we have this type 1 cluster here that is most closely related to a combination of North Atlantic and North Pacific types. And this cluster here, we only have a couple samples, but it's a combination of individuals from the North Atlantic. This deeper branch is one animal from Hawaii, and then there's the North Pacific residents and offshores. So the picture's not completely clear about the evolutionary patterns of, they didn't all evolve in their respective ocean basins. There's been movement back and forth in subsequent diversification. But for the most part, except in the North Atlantic, where we're still kind of getting our information, and I'm trying to get more samples, the ecotypes segregate out into these clear evolutionary units. I put this on there just to say that this tree, with the data that we have, the whole mitochondrial genome, is extremely robust. If you know about bootstrap values, it's kind of a percent from 0 to 100. These are basically 100. That's the posterior probability. Everything where there's a 1. All of these branches are highly supported, so this is a very robust tree. So, getting back to the technology, the next generation sequencing is this highly parallel sequencing technology of small fragments of DNA, where you generate literally hundreds of millions to billions of sequences in a single instrument run. Because you're generating so much data, it's actually more data than we need for these types of projects. These were developed primarily for sequencing whole genomes, where you're talking about 3 billion base pairs in a mammalian genome. But we're not interested in doing that yet for cetaceans. Well, at least that's not what our lab's going to probably be doing. But we want to generate maybe thousands or tens of thousands of base pairs for each individual, and we can multiplex samples. So we can do now up to about 120 individuals in a single run of an instrument, a single lane on an instrument. And the cost per, if you compare to what we got before, can you back it up for me? It doesn't seem to be responding. Still not responding. Anyway.
Tech support's on the way.
Oh, no, it's doing something. Okay. That just doesn't want to go back beyond that one. The last thing on that slide was that it was about, it used to cost us, or does cost us with Sanger sequencing, about $6 per 1,000 base pairs to generate mitochondrial control regions. With this technology, it's less than 5 cents per 1,000 base pairs. So the relative cost, and it keeps going down, which just goes down faster and faster. So there are many potential applications for this technology. We've been doing phylogeography and population structure using the mitogenomes. We're also using this for SNP discovery. So we're including up to about 50 nuclear genes on each array that we sequence. And so we sequence for 100 individuals, 50 genes, and discover nuclear variability that we can use to do genotyping on the nuclear side. Also, some new methods that we're using use capture arrays instead of the long-range PCR. So our DNA fragments can be as small as 100 base pairs, and so that opens up the ability to do poor quality integrated DNA, such as museum samples, even fecal samples. You can extract the DNA of the target individual, the cetacean, from the feces, and do whole mitochondrial sequencing and even nuclear gene sequencing. So it's not necessarily easy because you're still working with very poor quality and low concentration samples, but it has been done even for samples that are tens of thousands of years old, and we're mostly looking at samples that are going to be in the less than 100-year-old range, at least for the population studies that we're doing. So I wanted to just put this up at the end just to kind of overview where we're going with this in our own lab. And we've basically been developing some new technologies, capture arrays followed by next generation sequencing. And we're applying it to additional work on killer whales. We're doing fin whales, sperm whales, blue whales, beaked whales, spinner and spotted dolphins potentially, and also working with the marine turtle group to do population structure, the SNP discovery, and green and mother bacterials.